We could probably go ahead and get started. Hey, folks. Happy Monday. How's everyone doing? How's life? We're almost done. 3 more weeks, two and finals. So if you took a week 10 final for this class, then even fewer weeks. You did? Oh, perfect. Perfect. Um, so in terms of logistical announcements, hopefully everyone remembered that you had a lab due last night at 10 p.m. Dang, attendance is dwindling. It's so empty. But yeah, you had a lab due last night at 10 p.m. So lab 8 is released, and this is the 8th out of 9 total labs. So almost done. So if you can finish this one and the next one, you're done for the quarter, at least until the final. But that'll be little. Um, so yeah, logistics wise, hopefully everyone has already signed up for your final exam. If you haven't, like you'll definitely want to do that ASAP. Um, yeah, you wanna do it early so you can get a good time slot. Um, yeah, I guess any questions about logistics? So Lab 8 is out. It's due next Sunday or like this upcoming Sunday. And come to lab section if you want help. But any questions about logistics, concerns, comments, dreams, aspirations? No? All good? Okay, perfect. Okay, so yeah, so just make sure to get working on lab 8. Make sure to keep practicing for the final. Just like with the midterm, every question is going to come from the practice problems. I forgot to post the practice problems from lab 8 onto the practice problems thing, but I'll do that like later when I get home or whatever. But you have access to it because lab 8 is released now. So yeah, cool. Let's go ahead and jump over to my slides. So daily recap, what have we done? We've done a lot of stuff so far. Week 1 was kind of just introducing all the problems we want to tackle. Week 2 was de novo genome assembly, de novo genome annotation. Week 3 was read mapping. So if I sequence a new sample, how do I align those reads against the reference genome? Week 4 was consensus sequence calling and variant calling. So now that I've mapped my reads, how do I use those read mappings to reconstruct this new sample's genome sequence? How do I output the deltas against the reference genome? Week 5, we talked about multiple sequence alignment. So now that I've done this process for like 16 million sequences, what do I do with them? We talked about regular multiple sequence alignment. We talked about some heuristics we could use for like reference guided multiple sequence, stuff like that. Then week 6, we introduced a bunch of different models of evolution. And then we said, here's how we can use those models of evolution to infer a phylogeny. And remember, because those models of evolution were were time reversible, the trees that we get are unrooted. So then last week we talked about, okay, given this unrooted tree, given that I have some temporal information, given that I might have a reasonable outgroup, given whatever information that I have, how do I root the tree? So determine where on this unrooted tree was the actual ancestor. That tells us directionality of evolution. It also tells us, where evolutionarily the ancestor was. And then secondly, how do we then scale the branches to be in unit of time? So this was a problem known as tree dating. So from our models, the branch lengths are unit of mutations, or really expected number of per site mutations. So basically, number of mutations divided by genome length so that you can kind of compare against different species without worrying about genome length being an issue. But then how do I scale those to be unit of time? And we mentioned this was mathematically equivalent to determining the mutation rate for each of the branches, because mutations length divided by mutations per time gets you time length. So that's where we left off last time. Any questions, concerns, comments? We introduced a bunch of different methods. We talked about their pros and cons. Everything all good? Yeah. Okay, perfect. So today we're finally going to be tackling, I guess last week and the week before, we were tackling some of the questions we were asking in week 1. We were wondering, how does this virus evolve, and kind of what is the evolutionary history back in time of this virus? But then there was a lot of other questions we wanted to tackle about, like, given all these people in my population, what are the relationships of their transmission events? So can I figure out like what subgroups are closely connected in terms of transmission? That might help me inform public health intervention. So like if a bunch of people are connected and their overlapping metadata says they were at a party together, versus not very many people are connected who have metadata overlapping saying they traveled, then maybe that tells us what types of interventions we should employ. So today we're going to talk about a technique known as molecular clustering. So first, I'm going to do a little bit of epidemiology definition stuff. So I'm going to introduce the concept known as transmission clusters. I'm then going to introduce the concept known as molecular clusters and distinguish it from transmission clusters. Then I'm going to talk about how we can find molecular clusters from sequences. And I'm going to end by talking about how we can find molecular clusters from phylogenies. I think this set of slides is probably going to be another one lecture set of slides, unless there ends up being a lot of questions. So by the end of this, try to think about things you want me to do on Wednesday. And yeah, we'll probably have quite a bit of time on Wednesday for random free-for-all. But cool, everyone ready to go? Questions, concerns, all good? Okay, so transmission clusters. So a transmission cluster is a group of infected persons connected by transmission. So it could generally represent recent and ongoing transmission in a population. So the transmission clusters, these are maybe where the most recent growth of a transmission, like of an epidemic, are happening. So if we can identify transmission clusters, that helps us inform where we should target prevention efforts. So for example, if I have a big population of people that have tested positive for this virus, but like some of that population was way back in time and they're not really spreading rapidly, versus some of them are more like recent and closely connected, those might be where I want to target my prevention efforts, because that's going to be the biggest impact of who I can prevent from getting the virus. So another kind of terminology that I want to introduce. So the transmission cluster was just the group of infected persons that were connected by transmission, emphasis on group of infected persons, versus the risk network is a group of persons who are closely related, who are infected or susceptible. So in other words, you should think the risk network is kind of the superset. It's people that are closely related by infection and their partners who are uninfected, susceptible. The transmission cluster is a subset of the risk network. So it's just the people who are actually infected. And if we can identify transmission clusters, that might help us identify who in the risk network is most at risk of transmission. So, I can try to target the people who are already infected, but in some cases, I might want to target people who are most susceptible. So if you're in high school, you probably in whatever health class, you probably learned about contraception and stuff like that. Those videos that they made you learn about cost money to produce, and implementing them in schools cost money as well. So that's more of a preventative type of intervention that's targeting people who are susceptible, not infected. So yeah, those types of things. Who are the people that are most susceptible? What is their age group? What are their risk factors? What types of things are they most receptive to learning about or using? That's kind of the downstream stuff that we would do with this information. Okay, so how do I actually identify transmission clusters? The kind of biggest data source that we use is viral case surveillance data. So in other words, I can just look at how many people are testing positive for my pathogen in a given geographic area. And if I notice that there's a stark increase in the number of diagnoses in a specific population or geographic area, hey, those might be transmission clusters. Now, before I show the next slide, can anyone think of a confounding factor that might plague this? So let's imagine I'm an epidemiologist. I'm tracking case counts in different regions, and I see an uptick. One explanation is more people are getting infected. That's why more people are testing positive. Is there any other confounding factor that might cause this without necessarily? Maybe you and then you.
Is it test number, or is it test proportion, positive test proportion to test number?
Let's just assume positive test number. So just how many tests are?
Could be more people taking tests.
Okay. Yeah. Did you have? Exactly. Yeah. So the answer that was given was it could just be that more people are taking tests. So that's a very important confounding factor you have to keep in mind. And I don't know if folks remember from the early days of COVID, but in the early days, it was really hard to get access to COVID tests. And then all of a sudden, through technological breakthroughs, we were able to test a lot more cheaply, a lot more scalably. So it was a lot easier to get access. So what you ended up seeing, if you're just looking zoomed out at the population level, you see huge upticks in the positive cases, but it's not that COVID is spreading way more rapidly. It's just that now people have access to tests. So you just kind of got to keep that in mind when you're doing that type of data analysis. Another way of identifying transmission clusters is through what's called partner services, and also through what's called contact investigation or contact tracing. So basically, let's say that someone gets newly diagnosed for this virus. You can survey or interview that newly diagnosed person to ask who other people might they have interacted with. So for HIV, this is standard of care. If someone tests positive for HIV, you ask them hey, who have your partners been in the recent days or months? And then you might reach out to those folks to ask them to get tested. At UCSD, as part of the Return to Learn program, that might have been like people here's like freshman year maybe. If someone tested positive for COVID in the dorms, they would then reach out to the other dorm mates to ask them like, hey, can you take a test? Have you been feeling symptoms? Stuff like that. So basically use the newly diagnosed people to help find other susceptible people that are close by through surveying. So yeah, common for HIV and sexually transmitted infections, but also using COVID-19. So these things you could have been doing even before taking this class. So those are not necessarily as interesting to us. What we're interested in is molecular surveillance data. So basically, let's take a look at molecular biological data. In the modern era, that typically means sequence data, but it could be like byproducts of a virus. It could be like certain proteins or certain small molecules. It could be things that might show up in a person's wastewater through immune response or something. But for our purposes, we're going to assume molecular data is sequence data. So this is more so what we are interested in. How can I use molecular data, aka sequence data, either from the patient or from viral particles to try to surveil how is, how are these transmission clusters growing, or what are the members of the transmission cluster? Okay, does that kind of make sense? That's the setup of today's topic. We have sequence data theoretically. How do we use it to identify transmission clusters? Okay, so it seems like there's no questions, and I'm just going to make sure, yeah, the chat, there's also no questions. So now I'm going to introduce another term called molecular cluster, which you might be able to guess what it means based on what I just said, but let's introduce it and then distinguish it from transmission clusters. So a molecular cluster is a group of persons linked by molecular data, or in our case, by sequence data. And by linked, we mean they're very closely related in terms of their sequence data. So if we were to infer a phylogeny, they fall close together. If we were to just look at their viral genome sequences, they're very, very similar. So people who, from their molecular data, we can tell they're linked by transmission. So a molecular cluster is a subset of the transmission cluster. So in other words, remember, the risk network is the superset. We have transmitted, kind of diagnosed people, as well as their susceptible partners. The transmission network is just the infected people. And then the molecular cluster is just infected people who we also have sequence data from and are connected closely, like even closer than in the transmission cluster. Or maybe not more closely, but in general, we have their sequence data. So what things you have to look out for? Not every single person who gets infected gets their molecular data collected. So for HIV, it is standard of care to get your viral sequence collected. But like for COVID, a lot of people probably nowadays, they just do a nasal swab at home. If they test positive, they just don't go out. They don't usually then do a sequencing experiment to see like, hey, what is the genome sequence of this COVID virus? So not every single infected person gets their molecular data collected. So yeah, molecular cluster, it's only the people in the transmission cluster who got sequenced. And it's really, really, really important, just like with the previous scenario, to keep in mind the people that are missing from the molecular cluster. So in other words, people who either are diagnosed but not sequenced, so people that are in the transmission cluster but not in the molecular cluster, or people who haven't even been diagnosed yet. So people that are not even in the transmission cluster, but they're in the risk network. Does that kind of make sense? Okay. We're zooming by, so feel free to interrupt me if you have any questions at any point. So the first thing that we're going to talk about is if you gave me a bunch of sequences, how can I find molecular clusters from those viral genome sequences? And the technique I'm going to be introducing, this is actually what's currently the state of the art for molecular surveillance of HIV in the US through the CDC. The contract that we're on is still not cut, so hopefully it stays that way, but I'll introduce what we're doing, and you're actually going to implement a simpler version of exactly the same pipeline that we're running at the CDC. So yeah, how do I find molecular clusters from sequences? Imagine that I collect a viral sequence from two people, X and Y. There's kind of three scenarios that we can imagine. One scenario is, let's imagine X and Y were directly related in terms of infection. So either one of them infected the other, or there's some third party that directly infected both of them. So here, the notation that I'm using, d of xy, I'm referring to the genetic distance between the sequence I collected from X and the sequence I collected from Y. So the genetic distance between their viral sequences is going to be super, 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 super tiny. So like for COVID, it might just be like one mutation, or it might not be any mutations. It might be perfectly identical sequences if you're directly connected by transmission. If they were not infected but from the same person, but they were in the same general group of transmission events. So maybe like they're both in the same dorm, so maybe the same person didn't infect both of them, but maybe there was like a relatively small transmission chain starting from someone and then going to both of them. Their pairwise distance is going to be not as tiny as the first bullet, but still pretty small. Versus if X and Y were infected from completely unrelated sources. So for example, imagine school starts, it's the beginning of return to learn. People are traveling to La Jolla from all over the country or all over the world. Two people living in the same dorm that both test positive might not have gotten it from the same exact source. It might have been en route from their respective hometowns, and their sources might be completely unrelated, even though they both happen to be testing positive at the same place. So if they're infected from completely unrelated sources, their pairwise distance is going to be really big. So a general simple idea we can have is we can theoretically link person X and person Y if the pairwise viral distance is very small. And then what we can do is for all pairs of individuals, we can link them. We can place a genetic link between them if the pairwise sequence distance is small. And each of the resulting chains of genetic links will define a molecular cluster. So basically, we pick some threshold that we think is reasonable. We link people if their pairwise distance is below that threshold. And then all the resulting chains, those are our molecular clusters. So I'm going to do a simple example just to kind of highlight it. So here, let's imagine I have 4 people, and I've shown their pairwise genetic distances. And let's say that we use a threshold of 0.015, which happens to be what the CDC uses for HIV. I would look at all these pairwise distances, and the dashed line means I haven't placed a genetic link. This is just the pairwise distance. And I would see, oh, hey, these two are less than or equal to 0.015. So I'm going to place these 2 genetic links. And then the connected chains in the resulting graph, aka the components, represent my molecular clusters. Okay, so does that general idea make sense? Because you're going to have to implement it. Yeah, go for it.
Like when you do compare the 2 viral sequences, are you like comparing them against like a reference sequence, or?
Ooh, good question.
One's like closer to what you want, or I don't know.
Yeah. So the question was like when I'm comparing the viral genome sequences, like what is this distance with respect to? It's with respect to each other. So I can imagine actually, that's a great question. I was gonna do a little bit of a follow-up, and this is a nice segue to that. So imagine like person 1's viral genome is ACGT, and person 2's viral genome is ACCT. When I'm doing their pair, and maybe let's say like person 3 is like ACGA. When I'm doing their pairwise distances, I would say the pairwise distance between this person and this person, yeah, is 1 in a sense. I'm going to correct it slightly, but in terms of Hamming distance, it would be 1. But yeah, great catch. And then this person and this person would be 1. And then this person and this person would be 2, if we're talking about Hamming distances. For this specific application, you might remember that way back when I introduced the TN93 model, back when we were doing our rapid fire of evolutionary models, I mentioned that if I were to create a plot where the horizontal axis is, let's see, over here. Let's say the horizontal axis is number of mutations. Let me make that a little bit wider. And then the vertical axis is Hamming distance. You might remember that I mentioned at small values of number of mutations, because the genome is relatively long, 1 mutation is probably going to result in 1 Hamming distance. 2 mutations is probably going to result in 2 Hamming distance. But as I get bigger, I'm going to start accidentally mutating the same positions, and this curve is going to start kind of tapering off. And then does anyone remember what is this? It's kind of hard to do with my trackpad, but I don't feel like getting my tablet out. What is that line over here? Let's say in terms of percentage. Does anyone remember? 75%, exactly. Basically, as number of mutations approaches infinity, you've mutated both sequences to the point that they're effectively just random sequences. So they're going to converge towards what is the Hamming distance of two random sequences, which would be 75%. You have a 75% chance that, like, let's say that you pick one sequence and then you pick the second sequence just randomly. The first letter, what is the probability that the other sequence has the same first letter is going to be, sorry, a different first letter is going to be 3 out of 4. Do that for each individual position independently, it's going to converge towards 75%. So in practice, what we usually do is we don't typically just rely on Hamming distance. So when I introduced the TN93 model, I had mentioned it's the most generalized model where we can infer the model parameters just from sequences. We could calculate the Hamming distance, and then we can calculate the frequency of A, C, G, and T by just empirically counting them across the two sequences. And I can actually come up with a correction technique where if I calculate the Hamming distance of two sequences, I can basically, given my learned model parameters, use this theoretical curve to correct my Hamming distance into genetic distance, which would be number of mutations or proportion of mutations. So number of mutations divided by sequence length. Yeah, great question. So that was a long-winded response to that kind of.
Yeah.
Perfect. Yeah, no problem. Yeah, great question. Any other questions about this approach? So basically, given all the possible pairwise distances between my sequences, place a genetic link if a given pairwise distance is less than or equal to a given linkage threshold. And then the resulting chains of that graph represent my molecular clusters. Yeah, go for it.
Do you choose the threshold kind of like by looking at what happens? Like, what if there's just a strain of the virus that barely mutates and leaves everyone one cluster because there's
Oh, perfect. I was actually going to ask exactly that question. I was going to have someone poke a hole in this, and that is the exact hole I want someone to poke. So the question was, how do you pick the threshold? And, you know, hopefully by the time you've gotten to this class, you've taken a couple CS classes, and hopefully like magic numbers is a taboo thing that you never do in any of your programs. You never hard code any like magic numbers in your code. So I kind of casually, quickly mentioned, oh, let's use a distance of 0.015 as our linkage threshold. And I just kind of casually mentioned, that's what the CDC uses. But you should be like, where the hell did that come from? Why is it 0.015? That's a clean number. And as a computer scientist, if anyone ever tells you a clean number, you should be like, some human made this up to sound clean. There's no way that this was mathematically designed. So how did they come up with the number 0.015? My understanding is in like the early to mid 2000s when sequencing was becoming a lot cheaper, they basically had a lot of people that had HIV that they did survey data to see who actually is linked to each other. So they a priori, so in advance, they knew exactly which pairs of individuals truly were connected by transmission. And then they also calculated all pairwise distances, and you're going to end up getting a plot that looks something like this. So you're going to get a histogram where the horizontal axis is genetic distance, aka number of mutations, and the vertical axis is number of pairs of people. And you're going to get a plot that looks something like this, where this section over here is the people who were truly linked, and they're going to have really, really small genetic distances. And then this portion here is going to be the people that weren't actually linked. And their goal is just to find some threshold in the middle that hopefully maximally separates these two groups. So hopefully people that are truly linked end up having linkage using that threshold, have a pairwise distance below that, or less than or equal to that. People who we know weren't truly linked have greater than or equal, have greater than that threshold. How do they pick 0.015? They just kind of played and looked for a clean number. But great question. So in practice, there's a few different mechanisms as to why this 0.015 is going to be a crappy threshold. So one thing is, what part of the genome am I looking at? So for HIV, they typically look at, it's kind of a combo region. So there's the pol region of HIV, which stands for polymerase. And then there's the GAG region that stands, I don't know, it's some other protein, the GAG protein. So pol-GAG is like the region that they typically genotype. And they use 0.015 specifically for that region. But if I pick some other part of the genome, or like maybe the whole genome, there's no reason why 0.015 would be appropriate. The other thing that is an issue is, let's imagine that I have like the global HIV phylogeny. So let's say that HIV, the ancestor is somewhere in like the 1950s, but it was introduced or detected in like the early 1970s. So let's say this is like maybe the common ancestor is somewhere in like the 1950s or something. And then maybe over here, this is when it was finally actually detected, somewhere in like the 1970s. Now let's imagine that I have in the 2000s a group of individuals that are closely related to each other, that I say these people are a molecular cluster. Well, if just I wait for time to pass, let's say that I didn't sample them at that exact time. Let's say that I was slow and time passed, and I instead sampled them later in time, but the same exact 4 people. As time progresses, they're no longer linked. Now, maybe you would argue they're no longer a molecular cluster because time has passed, but I mean, technically, they were a molecular cluster. It's just that by the time I looked at them, enough time has passed, but they could have been infecting other people. So what part of the genome you're looking at, and then also time since their most recent common ancestor are both going to theoretically break that. Yeah. Seems flimsy, I know. Ooh, I like your idea. So you're going to see later on, how can we use a phylogeny to infer these? But for whatever reason, the CDC is using this. And whatever reason is phylogenetic inference is computationally intensive. So across the entire US, we have like 100,000 HIV genomes. It's technically feasible, but they want to be able to do this type of analysis in real time. So calculating pairwise distances, well, I could do multiple sequence alignment very quickly using tools like viral MSA. We actually use a slightly different tool that is codon aware, but that's beside the point. And then I can calculate pairwise distances by just doing Hamming distances. So keep track of number of mutations, keep track of count of A, count of C, count of G, count of D. Do a like just arithmetic to calculate the corrected genetic distance. You can do this super fast. So they use this kind of not super great approach because it's really fast and it's good enough, is the selling point. Awesome. Yeah, great question. Any other questions about that or any follow-ups? Awesome. Okay. So for your purposes, though, as the bioinformatician, some smart epidemiologist is going to say, hey, this is the threshold we want to use. And then you just kind of crunch the data and then give them back their clusters. Awesome. So this is implemented in a tool called HIV Trace. It's open source. You can find it on GitHub. The link is in the slides. And then, so this tool was published in 2018, but it was originally developed in 2013 at the start of the CDC contract. And then I don't have a picture of it. Dang it. I was brought into the contract last year, or maybe 2 years ago. I don't remember. But yeah, so this is kind of currently the state of the art for molecular clustering from sequences. Cool. Okay. Any other questions about that? Kind of makes sense. Awesome. Okay. So there's a lot of issues with getting molecular clusters from sequences. Is there some way of getting the molecular clusters from the phylogeny itself? So we went through all this labor in week 6 and week 7 to figure out how do I infer a phylogeny, and then how do I root and date the phylogeny. If I do that, can I somehow use that phylogeny to do molecular clustering directly on the tree? And just to kind of recap, to give ourselves like a little bit of kind of sense of how we might use properties of the phylogeny to do transmission clustering or molecular clustering. Let's remember the relationship between the transmission network and the phylogeny. So the transmission network, it's the true collection of transmission events. So it's a directed graph, specifically a subgraph of the contact network, where the edges have some time associated with them, representing the transmission events. And if I zoom in on a specific transmission event, there's some evolutionary history going on in this person where this time point at the top of this tree represents the time this person was initially infected. And then time progressed, and then the virus evolved in their body. And at the leaves, at the time of transmission, we have some viral diversity down here. And then the transmission event we can imagine is like I'm randomly plucking one of those viral lineages at present day, sending it into the recipient, and it's going to start a similar evolutionary process in this person at their infection time, while these sequences continue to evolve. So these continue to evolve. This has now spawned on its own evolutionary history, where the root of this subtree is the time of the transmission event. Okay, now I sample a couple leaves from each person. Maybe these are nasal swabs, maybe these are blood draws, but somehow I collect some viral particles, which are the leaves at the time of sampling. So the true observed phylogeny that I could hope to reconstruct is this observed phylogeny that doesn't have any of those purple unsampled branches. And yeah, so this is kind of the best that I can do. Okay, and kind of notice, so the branching pattern is going to be heavily constrained by that transmission network. So how can I do molecular clustering by phylogeny? So let's think about the same three scenarios, but how they're going to reflect in the tree. So the viral phylogeny, it was heavily constrained by the transmission network. If X and Y were infected by the same person, or like super, super, super directly closely related in terms of the transmission events, they're going to be really, really close in the phylogeny. They might be siblings in the phylogeny. They might be like in the same very tight subtree. They're going to be super close together. If they were not infected from the same person, but they're still from the same general group of transmissions, they're not going to be as close together, but they're still going to be really, really close. So they're still going to be relatively in the same general kind of part of the tree. Versus if they're infected from completely unrelated sources, there's absolutely no rhyme or reason for how they're going to be falling in the tree. They're going to be in arbitrary random points of the tree, so they're going to be super far apart. So as an idea, we could define clusters using relationships directly within the phylogeny. And this is a very vague term. How can I define those clusters? There's a bunch of different ways to define them. One option might be maybe I'm going to determine what are all the subtrees that have a maximum pairwise distance below some threshold, or maybe I decide to cut the tree in some way. So in the world of graph theory, there's various like graph cutting algorithms. So maybe I cut branches above a certain length, or maybe other ways of kind of cutting the tree. Or maybe just like with HIV trace, so the clustering method that HIV trace was using is called single linkage clustering, where basically you belong to a cluster if you have just one link to one item in that cluster. So what if I do the exact same clustering approach as HIV trace, but instead of doing pairwise distances from the sequences, I could calculate pairwise distances in the tree, just like you were doing on the previous lab, or maybe the one before, I don't remember. Either way. So in 2019, we published this tool called Tree Cluster. I wrote it, and then the other person did more of the theory behind it. Where basically, given a tree, it uses TreeSwift to traverse the tree and very efficiently implement whatever clustering methods that you want. So if you pull up the GitHub for this tool, there's a lot of different clustering methods that you could do. So you can maybe like cluster the leaves such that the average pairwise distance is below some threshold, and you're forcing the clusters to be complete subtrees. Or maybe you want the distances of the leaves to like from the leaves to be like maybe cut the tree some distance from the leaves. I don't know, all sorts of different ways of defining clusters. Maximum distance in a subtree, medium distance in a subtree, cut it some distance from the root, single linkage clustering, total branch length below some threshold, etc. So again, the epidemiologist would have to tell you like what is a reasonable way of defining clusters in this context and what is a reasonable threshold. But given some clustering approach and some threshold, now you can run this tool. It's going to cut your tree, output clusters that kind of group your pieces together in whatever way that they had defined. So in terms of performance, tree cluster is over here, HIV trace is over here. There was another tool that was using phylogenies to do clusters, to do clustering called Cluster Picker, but for some reason it's super slow. So yeah, this tree-based clustering approach, because you can define a lot of these clustering definitions as like a pre-order traversal or like a post-order traversal or just a couple different linear time traversal on the tree, it's super, super, super fast versus HIV trace will always have to do a quadratic number of pairwise distance calculations. So if you can be clever enough to define your clustering appropriately on a tree, then you can take advantage of linearity of tree traversals. Okay, that was a lot faster than I was expected. That was my last slide. Any questions about this, about any other topics from today's slides? People want me to like set up any of these tools? Do people want me to run any of them? We have like 15 minutes left. What's the best use of time? So yeah, go for it, go for it.
Like you have two sequences that are outside of the linkage. What was it called again?
Like the genetic linkage threshold?
Yeah,
Yeah.
sorry. You have something that's outside of that. What do you do with that data? Do you throw it away or?
Ooh, good question. So the question was like, let's imagine that, like, what do I do with the pairwise distances? Let's say I was doing the sequence-based clustering. I guess, are you asking for like individual sequences that are greater than the linkage threshold to all other sequences? Or do you mean like, okay, go for it, go for
Or
it.
like, let's say like the threshold you set on I think 0.02 or
Mhm.
something like that. Like all the other ones 0.05 or something like that. What do you do with those? Like what do you do with A to D that linkage?
I see, I see. So the question was like, what do I do with the pairwise distances that are bigger than my threshold? Depends what you want to do. So in terms of clustering, we ignore them. We just throw them out. And actually, the HIV trace pipeline, it as an optimization, instead of calculating every single possible pairwise distance to completion, the moment it calculates that you're about to exceed the genetic linkage threshold, it stops. Yeah, exactly. So if in implementation, you would theoretically early out. In practice, so as you mentioned, these distance thresholds might be somewhat arbitrary. So you might calculate all the genetic distance, like pairwise distances, and then do the clustering afterwards. So in lab 8, let me maybe pull up lab 8 real quick. Let's see. Because like it would suck if you like used one threshold and then like next week you want to try a slightly bigger threshold, then okay, crap, I have to recalculate everything again. So in practice, sometimes what you'll do, which is exactly what you're doing in 8, like problem 6 of lab 8, is you might be given every single possible pairwise distance. So here I'm giving you every pairwise distance. I have 5 sequences. I'm giving you all all 5 choose 2 pairwise distances. And then you can cluster on those. So if anyone took CSE 100, you implemented like smallest connecting threshold. That's effectively the same general idea where I can give you all the pairwise distances and then you can kind of ignore the ones above some threshold. Oh, actually, no, it wasn't smallest connecting threshold. It was the connected components piece. So connected components piece of 100 is exactly this. Yeah. And if you haven't taken 100, that's okay. Sneak preview. Awesome. Yeah, great question. Any other questions? I can also work on some of the randomly generated problems to show you how I would solve them. Just because you're still gonna have to resolve them yourself. Yeah, go for it.
For the transmission network directed as is it based on the sample time and the early one towards the later sample? Is that what it?
Good question. So the question was for this transmission network, the directionality, how do we determine it? So in terms of transmission network, and this is kind of a key distinction between transmission clustering and the transmission network itself. The transmission network is the ground truth history of what actually happened, which I might not have information about. So the transmission clustering process, and by extension, the molecular clustering process, it's a way of us using computational methods to try to reconstruct properties of the transmission network. If we could completely reconstruct the entire transmission network perfectly, including who infected whom and what is the directionality, that would be amazing. And there are some tools that try to do this. And as you suggested, one approach is if I somehow have figured out that these two people are directly connected with transmission, so like there wasn't a third party that's missing, but this person, I somehow have convinced myself definitely infected this person, you could use the infection times to figure out directionality because the person that was infected first must have infected the other person. The problem is figuring out this person and this person are definitely directly connected. That's a very hard problem that we're not sure if there's even a solution, because it's indistinguishable from this person infected a middle person who infected this person, or maybe there's a middle person who infected this person and then later infected this person. We can't really distinguish those. So given that we have missing data, it's kind of hard to disambiguate. So that's why we rely on clustering, which is non-directional, but still groups them together. Yeah, great question. Yeah, so here, this is kind of the ground truth. So we're pretending we're an oracle that knows everything. And then given that we know this everything thing, now how do we reconstruct, or at least reconstruct whatever we can? Great question. Yeah, any other questions, concerns? If not, I can just work on the prairie learn problems and you can all do them with me if you want. Sound reasonable? Okay, yeah, might as well. We have 10 minutes. So for the first problem, calculating Hamming distance. So Hamming distance, as I mentioned previously, we might want to define Hamming distance as a count. So I think that's how you implement it on a previous lab. Or you might or is that what you're doing on this lab? Oh, that's on this lab? Oh, perfect. Okay, so you're going to do it on this lab. So when you implement Hamming distance, you might define it as a just raw count, or you might define it as a proportion. So count divided by length of the genome sequences that you can directly compare two different Hamming distances. So this first problem, what is the Hamming distance between the following pairs of sequences? You just literally count how many positions are different. So in this, it seems like, it's kind of hard to see from the side. I think 2. It seems like, whoops. Unless I'm misreading it, I think just, oh geez. I think just these 2 columns. Yay. So yeah, you just count how many positions are different. That's not too hard. Then for the proportion, same general idea, but then you normalize by, so you divide by the total number of columns. So here, it looks like there's, let's see, so that's not the same. 1, 2, 3. So it looks like it's 3 out of 1, 2, 3, 4, 5, 6, 7, 8. So 3/8. I should be able to do that in my head, but I don't feel like it. Oh yeah, good catch. Yeah, 0.375. So we're entering it as a decimal, so 0.375. Whoops. Success. So yeah, so just count the Hamming distance, but then divide by the number of columns. And then genetic links, this one, the key thing is genetic links are less than or equal to. So if you have equal to, you should include it in your genetic linkage. So here, genetic linkage threshold of 0.024, which of these pairwise distances are less than or equal to that? 0.022, 0.01, 0.011, 0.02, and I think that's it. Awesome. So yeah, those first parts, not too hard. I don't want to give too much away for these other parts, so I'm going to let you maybe tackle them on your own. But yeah, maybe I can give an overview since we still have 7 minutes. Yeah, go for it. Go for it.
For the Unix one, the run TN93,
Mhm.
this is just like a general question, but it says here like at the end, it says your script should run it appropriately and create an output, and then towards the end it mentions containing space delimited pairwise distances. What is like space delimited mean?
Yeah, great question. So yeah, let me go ahead and open up the workspace. So the question was, while that's loading, so over here it's describing how you should calculate, like, I mean, this part over here is trying to give you some hints as to what parameters you should be giving TN93. And then over here, I'm specifying you should output your pairwise distances as space delimited. And the question was, what is space delimited? So in general, for plain text files, let me maybe open up my terminal. So for plain text files, like delimited just means if I have multiple columns, what symbol am I using to designate what's the separation between the columns? So like comma separated values, a CSV file, uses comma to delimit them. Tab delimited would be tab. So in this case, I'm saying the separation between the columns. So here, let me show you. Actually, let's just run TN93 real quick, just using the default settings. Then you're gonna have to look through to see how to change it. But TN93-H. So the input file, you just give it as the last argument. So let's say I run TN93 on example.aln. It has all this junk to standard error. Oh, let me just pipe it to less. It's bigger. So over here, and maybe let me make a smaller file. Let's see. So let me make a smaller FAST file. So sequence 1, let's say ACGT. Seq2, let's say ACCT. Seq3, let's see ACGT, and that's good enough for now. Just because the default is kind of hard to read. So let me make this big again. So my small alignment file, whoops, and let me open it and close it in Vim so that it adds the new, oh, it didn't add the new line. The heck? Ah! Oh, geez. Vim, small.aln. Oh, I don't have Vim, dang it. Oh, rookie mistake. I gotta add Vim to the image. I'll do that later. Okay, there we go. So now I have a small sequence alignment. Let me move that up a bit so everyone can see. Has ACGT, ACCT, and then ACGT. So I'm gonna do TN93 small.aln. It outputs all this. Let me just type to less. Oh, it didn't output anything. The heck? Should have outputted. Huh. Two of them should be identical. Why did I do that wrong? ACGT and ACGT. Seq1 and Seq3 should be identical. That's weird. Whatever. I'm just gonna go here and change these names to smaller names. Seq1. Oh, that's gonna be pain. Maybe not. Okay, I give up. So key takeaway is the columns in the TN93 output are separated by commas. So by space delimit, it just means separate these columns by spaces. Long-winded answer. I thought it was gonna be much shorter. Sorry.
Stylistic, or is it like actually used for something?
Great question. So the question was, is it stylistic or is it actually used for something? So let's run TN93 on the original example file. And you'll notice that the oh, I guess these names, yeah, so the names, actually I could just open it. So the names themselves have commas, which if TN93 was a little bit more clever, which I should tell the creator to fix it, he's my colleague, but in general, a CSV file, you should wrap commas, like if the strings themselves have commas in it, you should wrap it in quotation marks. But since he hasn't done that, technically this file is malformed because this comma that's part of this genome sequence name would be treated as a separate column, which kind of sucks. So by designating what we're going to delimit by, it's helpful to like make sure that we don't have any conflicts with, yeah, just basically make sure there's no conflicts in how we interpret the columns. Yeah. Great question. Any other questions? And it's going to be one of the command line parameters. So you could just take a look at the help message, and basically each of the different things I'm asking you to change is going to directly map to something in the usage. So hey, delimiter, huh? That might be useful. Yeah. You could do that too. So yeah, you could theoretically. Oh, if you. Oh shoot. Yeah, I didn't include any. You're lucky. You got lucky. So my grader doesn't include commas in the names. Otherwise, it would have broken if you just output their.
On the documentation that capital T for.
Like on the web documentation.
I think it was I found it in the README, like example usage.
Oh, that was.
Yeah, so I think he added that feature. Let's take a look. So that's HTML3. I think. Let's see. Oh yeah, there we go. Yeah, so I think he more recently added that. So yeah, that was my pain point, and Sergey fixed it. So I think he probably just kind of quickly fixed this October 20th, October 21st. Yeah, I think he just quickly fixed it and then didn't update it everywhere in the documentation. So I think this thing he just forgot to update. Whoops. So now let's learn some good software engineering. Oh, what the heck? There we go. Okay, copy that. And this will be the last thing that I do. So he starts with the optional arguments. So maybe I should start there. Whoops. Optional arguments is over here. There we go. And then I think I put an extra. Let's see. Show diff. Oh, geez. Did I break it? Whatever. Okay, we won't be good Samaritans. But if someone wants to, you can feel free to make a pull request that has those changes added to it where you fix the usage message. But I don't feel like doing it. Okay, cool. So let's go ahead and call it there. So yeah, so hopefully now you know how to solve the first three problems. The last three, they're going to be a little bit more tricky, but I think you'll be able to do them. Not too bad. And yeah, come to lab section if you want help. Awesome. Thanks so much, folks. Any questions from folks on Zoom? I guess people on Zoom already kind of left. Questions? Nope. Okie dokes. Awesome. Yeah, thanks so much.